Hi guys. So this is the first in a series of videos I'll be making discussing biochemical pathways. And in today's video, I'm going to be talking about the urea cycle. So what is the urea cycle? So in mammals, excess nitrogenous waste is packaged as urea and excreted in urine. And urea is synthesized from ammonia. So here I'm showing ammonia in its protonated state in physiological pH. So this process, the urea cycle, happens exclusively in the liver, and I'll probably talk about it later in other videos. The liver is this specialized organ that's capable of doing a lot of these special processes. And so the question becomes, how do we transport the ammonia into the liver? Uh, because as it turns out, ammonia is toxic. So we actually can't just transport it in the blood to the liver as you would think. So the body has these very interesting mechanisms to bypass that problem. So in this introductory video, I want to discuss two of those methods. So the first of these methods is actually using the amino acid glutamine. So glutamine can actually safely transport excess ammonia to the liver. And in the liver, it can be acted upon by this enzyme called glutaminase. So glutaminase mediates the hydrolysis of glutamine to actually liberate the amine group, so liberating the ammonia. So that's one way of doing it. The second way is a little bit more involved. So in muscle tissue, metabolically active cells will naturally produce pyruvate, a metabolite of glycolysis. And this pyruvate will interact with another amino acid, glutamate, in this really cool reaction mediated by this alanine transaminase. So I'll probably make a very detailed video about these enzymes, but there are dozens of transaminases, and they mediate this really cool ping pong reaction where the carbonyl of a keto acid is essentially exchanged for the amine group of an amino acid. And in this particular transaminase reaction, the products here are another amino acid, alanine, and another keto acid, alpha ketoglutarate. So this alanine now can freely be transported in the blood to the liver. And in the liver, it's going to be acted upon again by alanine transaminase to again spit back out glutamate. And now glutamate is going to be acted upon by this enzyme, glutamate dehydrogenase, to finally liberate our ammonia and deposit it into the liver. Uh, Now, you may be asking, why don't we just transport the glutamate directly to the liver? And the problem there is that glutamate is actually a potent excitatory neurotransmitter. So there is a problem in having an abundance of glutamate in the blood. And also, it's a precursor for another neurotransmitter, GABA aminobutyric acid, GABA. So overall, the body has this very clever mechanism of circumventing that problem. So in the next video, I'm actually going to talk about the enzymes involved in the actual urea cycle.